Good morning, everybody. Hopefully everyone can hear me loud and clearly. Someone could chat something encouraging into the chat box, that would be fantastic. Just to let me know that I'm coming through. Excellent. Welcome Desiree, Greg and Nick. Thanks for coming on board. We've had quite a number of registrations, relatively recently as well, so I think we'll have a few people slipping into the webinar as we get going. It will also be recorded and available to all registrants as well. It's fantastic to have you on board. Really excited about this webinar, and it's a real privilege to have Dora Taggert with us today, who's the president of Microbial Insights. Dora's got a long history in using sophisticated molecular tools for environmental diagnostics, from site remediation through to corrosion diagnostics, and even tracking contaminants from particular water sources, for example. What I particularly like about Microbial Insights is that they are really quite sophisticated tools, but they're highly accessible for environmental scientists and contaminated land managers in the field. So without further ado, I'd like to welcome Dora to this webinar. Thank you very much for coming, and look forward to hearing what you have to say.
Thank you very much. Thank you, Alistair, and thank you everyone for taking time. I know you're all busy, so hopefully you'll find this to be a useful amount of time that you spend today. During the presentation today, I'm hoping that I can cover some basic information about different molecular tools that are available for us in site management, talk a little bit to you about how we can use these tools to help us make more effective decisions at sites so that we can be as cost conscientious as possible. Also, for any of you on here that may be more advanced in the use, I've included a few tidbits about some upcoming information and projects that hopefully will provide some new information and be valuable to you as well. Okay, I'm not going to spend a lot of time on this slide. I think most of you are here today because you understand the importance of using this third line of evidence that microbial data can provide. You know, often we get our contaminant concentrations and we see that the source area versus downgrading in our plume, there's differences in the contaminant concentrations. The geochemistry, we look at that and we see that there are changes in the geochemistry, that maybe our electron donors or acceptors are being consumed as we move through the plume. But often with that, we're left with an incomplete picture where we put stuff in the environment or made changes, but we've still got that black box of actually what's happening in the subsurface. So I think these tools are a way that we can address that black box and try to understand better what's happening with our microorganisms. And in the past, the way that we would look at different microorganisms is we would do plate counts. And I don't know if any of you have done these back in college or graduate school, but for those of you that have, I'm sure you can attest to the fact that they're difficult to get to work appropriately, like Alistair's comment there. They're awful. We just can't replicate in situ conditions in the lab, no matter how hard we try. So there's been a lot of publications that have come out, people talking about the difficulty of growing these microorganisms. And we know from this that less than 1% of bacteria can actually be cultured in the lab. Either we simply don't know how to grow them, or we need a consortium of these microorganisms for them to grow efficiently. So we just can't get it right in the lab. So with these molecular tools that have come out, we can directly assess our microbial populations by looking at the DNA or the RNA or the proteins of these organisms, so we can get a better understanding of what they're doing. The electron acceptors are being consumed. This particular organism is present. That's likely what's happening. So we can get more of a direct link to our microbial community and to the organisms that are actually doing the actions that we want out there in the environment. So what I hope you walk away with today is thinking about these different tools. And there's always new exciting technologies that are coming out. And to be honest, the nerdy scientist in me gets excited every time we have a new discovery and we can find a new way to evaluate these microbial populations. But from a practical standpoint, that we got the question we're trying to answer at our site, and then pick the molecular tool that answers that question. So when I was putting this presentation together, I was thinking about what those questions are. So often we have questions like, what's the concentration of our contaminant degraders? And we can use tools like quantitative PCR or Quantaray to help us answer that information. So these are tools that we extract the DNA from our sample, whether it's a groundwater or a soil, and we use this analysis to give us a number of a particular microorganism or a particular functional gene that we're interested in. Another question that we might ask at sites is, show me that degradation is actually occurring. So there's some tools that have come out within the last decade that are really exciting. There's compound-specific isotope analysis. Some of you have probably used that at your sites where we look at natural fractionation, and we're going to talk about that briefly during the presentation today. Another tool that I really love that gives a very clear answer is called staple isotope probing. We'll go through that and how it helps us prove biodegradation. A newer tool that you might have heard a lot about from human genome sequencing and what's going on is next generation sequencing, looking at metagenomics and trying to understand the microorganisms that are present at our site. And this is a really valuable tool that we can use when we're trying to understand maybe a new contaminant where we don't know a particular degradation pathway that we want to target. It's a great tool to be able to look at our microbial community as a whole. And then finally, a tool that I really like that I think is very appropriate and cost effective is called an in situ microcosm. It helps us select our treatment strategy. It's similar to a bench scale study, but we're actually doing it in situ in the field. So I'm going to cover some of those topics today. And we have a small audience here, so if it's okay with Alistair, what I'd like to do is just have people stop and ask me a question if there's anything that's confusing. We can take our time and go through each of these tools and answer anything. And then if you guys have questions after, I'm happy we can set up another webinar that we could go more into depth about project or particular tool or a product that you're specifically interested in. Okay, so the first thing that I want to talk about, and it's probably a tool that you've heard about and some of you may have used, is called qPCR or Quantaray. So qPCR is a technology that we use that's been well vetted as the most precise way that we can accurately quantify specific microbial populations or genes that we're interested in. So with this type of technology, what we're going to do is either look at a taxonomic identification so we can learn who a microorganism is. For those of you that have worked on chlorinated solvent sites, you've more likely heard of Dehalococoides. A really important organism for us to understand. So if we do a taxonomic identification, we can quantify the number of Dehalococoides there, and I'm going to show you an example of this. Another thing we might want to do is look at a functional gene, because there are times that we don't care who the microorganism is. For instance, anaerobic benzene degradation. I don't really care the name of the person doing it. There can be a lot of different microorganisms that have this capability. I just want to know, can they degrade that benzene? Do they have that gene? So in those cases, we'll look at a functional gene, either a reductase or an oxygenase gene that's involved in that degradation pathway. So the way that this works is with qPCR, we're going to extract the DNA directly from the microorganisms in our sample. We're not going to culture anything or try to grow it on plates. And then we're going to run individual assays to be able to target all of that particular gene or all of that particular organism that we're interested in understanding. So at the end, you're going to get a quantitative number, so many cells per milliliter of sample or per gram of our soil. But because some sites and processes can be more complex, there can be mixtures of contaminants, or for some contaminants, multiple pathways. So we have a tool called the Quantaray, which I like to think of like qPCR on steroids, because it allows us to target many different pathways and organisms all in a single analysis. So as with qPCR, the DNA is extracted and it works exactly the same way. What we'll do is extract the DNA and then we apply it to the slide like you see here in this picture. Hopefully you can see my pointer there. Each of those little squares is a sample, and inside those samples are these little holes. And each of those holes is like those tubes you saw in the previous picture. And we are going to generate curves that we can correlate back to known concentrations of that gene. So rather than getting a single analysis, you're going to get a lot of different targets that you're interested in for a particular contaminant. So we can get a lot of different pathways, and I'll talk about those in just a moment. So one of the questions that we have is, how sterile does the sampling have to be? Well, you want to be as representative of your aquifer as you can. So what we would do is send you sampling supplies that are sterile, and HydroTerra has those in stock there. And you would just want to use gloves in the field and do the sample collection. You know, it's pretty much the same as what you would do for your chemistry and geochemistry, those same safe handling techniques. And especially with like the bioflow filters that we have, the little encapsulated filters that you put water through, those are very easy to use in the field. And to be honest, they're almost impossible to contaminate. So I think you'll find sampling isn't very difficult. And then you just put it in a cooler and ship it to our lab there in Adelaide. The next question that we have is about hold times, and that varies. We like to get the samples as quickly as possible, but the key point in all the studies that we've done is to keep the sample cold. So if you can get the sample on ice so that it stays close to 4 degrees, then that's really the key factor to minimize changes in your microbial community. When those cells are at that temperature, activity really slows, so it minimizes any changes in it. We recommend that you try to get it to our lab there in Adelaide within 24 to 48 hours. But again, we know that occasionally there's shipping concerns. That's why we take the temperature of every cooler that comes in. So the critical point is just making sure you use enough ice to keep it cold during the transit. Good questions. Okay, so we talked about the Quantaray, and we want to use this. And I love this tool, especially when we're talking about petroleum hydrocarbon sites, because with petroleum sites, we can have a lot of processes going on at the same time and a lot of a mixture of contaminants. For instance, we may have aerobic BTEX and MTBE, PAHs and alkanes that are present, and there's a lot of different organisms and functional genes that can be important. So if we ran all of those qPCR assays, the cost would be very high. But with the Quantaray, we can do it for a much more economical price, and that way we can look at all these pathways and not have to pick and choose which we're going to do. So with the Quantaray Petroleum, we're going to look at both aerobic and anaerobic pathways to be able to understand the degradation of our contaminants. So it can give us a really good understanding of what's happening at that particular site location. One of the questions I get a lot is, where should I collect the samples? My recommendation would be that you do a cross gradient, do your source area, and then where you see changes in the concentration of the contaminant, or if you see geochemical changes where a certain area is a lot different than other areas of the site. Maybe there's a lot more sulfate in an area, or it's very depleted in one of the locations, or maybe the down gradient is more aerobic. So you want to pick locations based on what you know from the past history of that site. For most sites, I would say it's an average of 3 to 4 samples, unless it's a very large plume. Okay, so we can do the same thing with the Quantaray, not just for petroleum hydrocarbons, but we can also do it for chlorinated compounds. We can have reductive dechlorination that's happening, looking at competitors for when we add our electron donor. So anytime we add an electron donor into the environment, for instance, a vegetable oil, that's going to be fermented, release the hydrogen. We want to stimulate those organisms that can do reductive dechlorination, like Dehalococoides. But we can also have sulfate reducers and methanogens that will use that available hydrogen. And we often see that there might be a little bit of a lag in the time where the sulfate reducers and methanogens become very active and utilize a lot of the hydrogen. And once they start to stabilize in their growth point, then you see reductive dechlorinators start to grow more quickly at the site. So it's kind of common, but this test allows us to be able to take a good look at that. The other thing that we can look at with the Quantaray chlor analysis is co-metabolism. And this is something that I feel we often miss at sites, looking at these pathways, because it's a little difficult, and in the past it's been more difficult to interpret the information. There's a lot of different functional genes and organisms that can kind of fortuitously degrade some of these compounds that we're interested in. So this may be happening while they're doing other processes. But we can still look at these genes, and I'm going to show you guys some information that's not really out yet, but is coming out. Dr. John Wilson, who just retired from the US EPA a couple of years ago, did a webinar for us on Tuesday. It's on the resource section of the MI website if you're interested. And he was talking about co-metabolism of chlorinated compounds and the research that we've done. So I want to linger here for just a minute and show you some of that data that John Wilson and Todd Wiedemeyer have come up with and been doing for some CERDIP funding, and give you kind of a sneak peek at this data. So you might want to listen to his webinar for more information, or if you're going to be coming to the Battelle conference in the US in Miami in May, then I would encourage you to come to the session that John Wilson and Todd Wiedemeyer have, and they're going to be going into this a lot more in depth and showing more data from this project. But this is kind of a sneak peek for you. So what we have here is graphed data from sites where we have TCE degradation rate constants, and we've compared the functional genes that are associated with co-metabolism. For instance, in this graph, I'm showing you PHE, which is a phenol hydroxylase gene. So if we focus on the data where there's significant amounts of degradation, those are our red dots on the graph. When TCE degradation is below this line, what we see is that PHE is also low or non-detect. When there's some degradation, in this middle box here, we see that PHE concentrations are around 10 to the 3 cells per mil. But finally, with faster degradation, we start to see higher concentrations of PHE, above 10 to the 4 cells per mil. So this isn't a gene that if you'd asked me a year ago, I would have expected to correlate so closely. I expected our SMMO for our methane monooxygenase, or perhaps a toluene dioxygenase gene that has a real relaxed specificity, to be the one that would correlate better. But we're finding that this PHE gene may be really important. So I'm excited about the lab studies that John and Todd are doing, and they're also expanding this to other compounds, not just chlorinateds, so we can start to understand other co-metabolic processes in the environment. And I think this could be really important for us to understand what those numbers mean. So again, I'd encourage you to either listen to that webinar or attend that session if possible. If you'd like more information, you can also email me and I'll put you in touch with them so that you could get more information on anything with that project. So just a little sneak peek for you. Okay, let's move on to our case study. This is a typical chemical facility with a mixture of chlorinated ethanes, ethenes, and propanes. So as I mentioned earlier, we always want to start with the questions that we want to answer for our site. As a side note, Microbial Insights has been processing qPCR samples since 2001. So we have a very large database of samples, a large history of this from around the world. And when we look at more than 13,000 samples where we have both Dihalococoides concentrations and all the chemistry for the site, we see that when Dihalococoides concentrations are above 10 to the 4 cells per mil, we find good rates of complete degradation. We see ethene formed about 80% of the time. So good odds, a good indicator for site management. So we can use this number to help us put into context our Quantaray data from that chlorinated site that I was just talking to you about. So what I'm showing here are dechlorinating organisms that are involved in the degradation of chlorinated compounds at one well from the site. I'm just focusing on that because of time today. This is our baseline sample. And we see that we have low concentrations of Dihalococoides, our DHC here, as well as 2 other organisms that are involved in reductive dechlorination, partial reductive dechlorination, Dihalobacter and Desulpheromonas. So when we look at the database rankings, we see that we're at the bottom 25th percentile from samples that we've analyzed around the world, and that our Dihalococoides is below that 10 to the 4 mark where we want to be. So unless we do something, we're probably not going to get really efficient rates of reductive dechlorination happening at this site. So what the site manager decided to do was to go in and do an injection and add an electron donor to the site. What I'm showing you here is the sampling after post-injection. It's those red lines that we're seeing there. And here we see organisms again involved in the reductive dechlorination of chlorinated ethanes, and propanes. And look at the change in those concentrations. We've now gone from the bottom 25th percentile to the top. We're at 90 to 98th percentile compared to samples around the world. So let's look over here at our Dihalococoides, and this is a vinyl chloride reductase gene found in some strains of Dihalococoides. So just post-injection, we're already almost to that 10 to the 4 mark, and we're now at almost 70% higher compared to other sites around the world. We also see that vinyl chloride reductase gene is now being detected. So with this Quantaray data, we can also start to see what may be causing this case. So in this plot, what I'm showing you are our total bacterial numbers, EBAC, and then I just talked about those sulfate reducers and methanogens that compete for that available hydrogen. So post-injection, we see an increase in both of these organisms, and this is commonly what we see at sites. But once the bloom in these organisms stabilize, we commonly see reductive dechlorinating populations start to increase. Typically, we'll see a 3 to 9 month lag depending on the sulfate concentrations at the site. So by having this Quantaray data, this can help us to link together what's happening. We can understand the reductive dechlorination. We can start to put together what may be causing lags in our reductive dechlorination and give us a lot of really helpful information. So as you've probably seen with data from several sites in previous presentations, qPCR and Quantaray can be applicable for both petroleum, hydrocarbon sites, as well as chlorinated solvents. These tools are applicable with all enhanced remediation products, whether you're talking about adding an ORC, EOX, if you're adding vegetable oils or other electron donors to the site. I recommend using them in the beginning if it's at all possible so that we can have a baseline concentration, just like we saw in that previous case study, really gives us a better idea of what's happening post our injections. And then kind of monitoring that over time can give us that direct evidence as to the effectiveness of our electron addition. qPCR and Quantaray are also great tools that we can use when we're talking about adding an inoculum. So if we're doing some type of bioaugmentation at a site. And then if we're adding activated carbon products. I've been really excited about some of the results that we've seen here in the US with some of these colloidal products that are being added into the environment. We're seeing some really enhanced degradation using qPCR and Quantaray. We can use this in combination with the chemistry and the geochemistry to help us better document and understand those tools. So this is a quick example of an injection of an activated carbon at a TCE impacted site. Here I've shown the contaminant concentrations as a function of time just before and after the activated carbon and electron donor injections. The TCE concentration, as you see, dropped very rapidly from 500 micrograms per liter to non-detect. So very effective absorption. That's what we'd want to see from this product, right? But what about biodegradation? After injection of the activated carbon, biostimulation with an electron donor and bioaugmentation, the Dihalococoides concentrations went up to greater than 10 to the 4 cells per mil, that critical concentration that we want for effective biodegradation. Dihalococoides was even maintained at greater than that number for almost 9 months, even after the removal of the chlorinated hydrocarbons from the dissolved phase. After this period, available electron donor concentrations, as indicated by the TOC that we're showing here in this graph, this line, began to decrease. By 15 months, TOC had decreased to less than 10 milligrams per liter, and Dihalococoides populations also decreased by approximately an order of magnitude. And I didn't show it here, but the vinyl chloride reductase concentrations that had been more than 10 to the 3 cells per mil decreased to only about an order of magnitude, about 10 to the 1 copies per mil. So the first thing to notice here is that I rescaled the y-axis so we could take a closer look at that daughter product concentrations. So what happened when we look at our daughter products here? Electron donor was completely consumed. And unfortunately, I don't have Quantaray data from this period, but if we assume that the observed trend continued, Dihalococoides probably continues to decrease. That makes sense. Doesn't have enough electron donor to support growth, redox conditions become less favorable. We see ferrous iron decrease, sulfate increase, and a return to less anaerobic conditions with consumption of the donor. Once dihalocopoides decreased, vinyl chloride was detected at very low concentrations. So this provided additional convincing evidence that reductive dechlorination of TCE had occurred when those conditions were right. So the takeaway from this study is that some of these activated carbon products are new and we are trying to understand the effectiveness in environmental applications. But this activated carbon combined with electron donor and bioaugmentation really resulted in effective absorption and biodegradation. Dihalocopoides is an obligate halorespiring microorganism, and there's no way that a DHC population of 10 to the 4 would have maintained for 9 months if there wasn't some type of activity that was happening there. Eventually, vinyl chloride was detected at very low concentrations, providing evidence of sequential reductive dechlorination. And it's important to remember that vinyl chloride was only detected once dihalocopoides concentrations decreased due to limited donor availability. So with additional electron donor or a second application, vinyl chloride concentrations, even very low ones, probably wouldn't have been detected at all, which leads me to my next point. Microbial monitoring, whether you're doing qPCR or Quantaray, is really important in these cases to help us understand biodegradation after these types of injections, because based on these results, we may not see the daughter products that we would sometimes use to help us track and understand the contaminant degradation over time. So that's just a quick overview of that. There's a lot of studies that are being done by some of these product vendors that I'm happy to share with you if anyone has questions about that, about these new activated carbon products. But I think it's really interesting, and combining with these tools, we can really figure out better, more effective treatment strategies that we can use. Okay, so are there any questions kind of focusing on qPCR and chlorinateds before I move on to the next topic? Okay, so the next thing that I want to cover briefly is called next generation sequencing or metagenomics. But because of time today, I'm not going to spend an enormous amount of time going into the details of this, but I did do a webinar a couple of months ago that talked about these tools more in depth and showed a lot of the data from this. So I would encourage you to go on and watch that webinar if you'd like additional information. So when people are talking about next generation sequencing, typically what they're referring to is community profiling using amplicon sequencing. This is where we take primer sets and we target the 16S gene that's found in all bacterial cells. So with this gene, we can get the name of the microorganism. Like I just talked about, when we name a microorganism like dihalocopoides, that's what this gene is going to tell us. So all it tells us when we do this type of technique is who is there. So with this type of analysis, you're going to get a chart similar to what I've shown here at the top, listing each sample analyzed and the number of reads that were sequenced in each sample. Then we can compare between samples to help us understand the difference in diversity between our samples at our site. Also, for each sample, you're going to get a bar chart like I've shown here. I've just shown one as an example for you, but it's going to show us how much we could identify the sequences that we detected in our sample. And typically, when I'm looking at the data from this type of analysis, I usually look at the genus level identification because I think for our purposes in bioremediation, it gives us the most useful level of information. So what you're also going to get for each sample is a table like the one I'm showing here and a pie chart showing the different genera that were detected in each sample. This table can give us some important generalizations about each of the different organisms that we've detected in our sample. So we can get some information like these may be anaerobic microorganisms, or some of them can do a particular degradation process. So it can give us some useful information in this chart. Also for each sample, we can do statistical analysis to help us better compare the microbial populations that are present at our site. I'm just showing you a quick example of that here. This is hierarchical clustering analysis that we can do with our statistics, and we can see which samples are most similar to each other. Then we can take a look more in depth and try to figure out what the common factor may be between these samples. For instance, you can see here this line that we've drawn down the middle. This is where the biggest division is between our two sample sets. And we can see that one is post-treatment, one is baseline. That's showing the difference between these groups. We can then use additional statistical analysis, such as principal component analysis, to help us understand what genera cause the organisms in those samples to group together. So we can go into this more if anyone has questions about it, but each of these dots would be one of our samples, and you can see they pull together, and what genera of organisms were detected here that weren't detected here in these samples that are further apart from them on this graph. So it can tell us what may be most important. Like in this group, Dichloromonas is pulling this group of this group of samples together from the microorganisms that were detected. So we can start to see what's causing the differences in our samples and give us a more depth of knowledge there. So I would like to encourage you that I think NGS is a great tool for exploratory questions and community profiling. And as I mentioned earlier, especially when we're talking about an emerging contaminant where we where we can target specific microorganisms, we don't know the pathway, this is a great tool for us to look at that total community shift. We can also use it when we apply a bioremediation product to see if we make things more anaerobic. So we can combine next generation sequencing and qPCR. These technologies are very complementary, and it can be very effective for us in site management. And as Alistair points out there, I think that PCA is a very powerful tool, and it's really simple once you look at it. I think there's an example in that webinar that I did that I'd be happy to send anyone that kind of talks about how PCA casts shadows on our samples, so we can get a better understanding of what's causing and what's the differentiating factors within our sample. It's, as he says, a very intuitive tool that makes interpretation much easier. So are there any questions about next generation sequencing? I just wanted to give you a brief overview because I know that people are hearing about that more, and I think it has its benefits and limitations that we need to make sure we understand so we can truly utilize that tool appropriately at our sites. Okay, so moving on, these are kind of my favorite tools that we're going to end with today. I want to talk about stable isotope probing and CSIA analysis. Again, with our time limitation today, we can't really get into the depths of the tools, but there's a great webinar on the MI website that Dr. Carrie Sublette did that talks about the differences between CSIA and stable isotope probing. So if you have time, I'd encourage you to, and interest in these techniques, I'd encourage you to watch that webinar. So let me go through this briefly for you. Let's get started by looking at CSIA. So before I get started, I also want to point out that there's an excellent guidance document that the US EPA put together on CSIA. Dr. John Wilson was one of the authors of this document. I would encourage you, if you're doing any CSIA analysis, to download it and use it. It's a very powerful document that gives you a lot of guidance. So if you're using CSIA for source delineation, or if you're using to understand degradation over time, and these different isotopes, which ones are most appropriate for which chemicals. This is actually a very, very good guidance document that spells it out very clearly for us. And I think Alistair is going to send you a link around about this document that's available. Okay, so before we get into this, we need to understand the reporting units that we're going to see, and this is the same unit of measure that we're going to get whether we're talking about stable isotope probing or if we're talking about CSIA analysis. It's called a delta value, and simply it's looking at the ratio of 13 carbon in our sample to 12 carbon in our sample versus that of an internationally recognized standard. So the reason that we do this is that we want to be able to look at sites around the world and be able to standardize that number so we have a unit of measure that we can always relate to so that we can compare samples. So that international standard is PD belimnite that's used around the world no matter what lab is doing this analysis. So before I dive into that, I just wanted to kind of explain that value in case you haven't heard that before. So let's talk about why CSIA works. So in nature, we know that there are two stable isotopes that we commonly use, a carbon-12 isotope and a carbon-13 isotope. So the chemical bonds with the lighter isotope, or carbon-12, are slightly weaker than those that are formed with our heavier isotopes, so they're going to react more quickly when degradation happens. So as a compound is biodegrading, that parent compound becomes enriched in the heavier isotope. There's going to be more of those 13 carbon bonds that are present. So we're going to see our ratio of 13C to 12C change. The daughter product is, when it's formed, is initially very depleted in that heavy isotope, but then it's going to become more positive as it's being biodegraded as well. So if we look at this, I borrowed this slide from Professor Mike Hyman at NC State because I think it does a great job of illustrating this concept. This is 13C chocolate fractionation. In this illustration, each M&M represents a molecule of a contaminant. If the carbon-12 is our blue and the carbon-13 is yellow, we go from left to right. The contaminant, our M&Ms, are being consumed. Very popular candy for me. So as I'm consuming them, let's say that I have a preference for blue M&Ms. So as you look from left to right, you'll notice that the overall number of M&Ms is decreasing, but the proportion of yellow to blue is increasing. That's analogous to what we see happening with the biodegradation of our naturally occurring contaminant. 13 carbon content in the undegraded pool is going to increase over time. CSIA has been used successfully over the past 5 to 7 years to really help provide a line of conclusive evidence of biodegradation. And I think you'll see that very clearly in that document from the US EPA. It's applicable to a wide variety of compounds, from chlorinated ethenes to 1,4-dioxane. And it's a great tool that we can use to understand the extent of degradation of our parent compounds at the site. It's also one of the few tools that we can use to help us understand abiotic degradation. So if you're adding ZVI or other iron-bearing minerals, this is a great tool to use to help you understand how much fractionation is happening. And to get this amount of information, it is relatively inexpensive. It's also a tool that we can commonly use to help us in our site management decisions and help us differentiate sources. So it's one of the few forensic type tools that we can use for source identification. So, I really like CSIA. I think there's a lot of value to it. Again, when we're talking about CSIA, we're looking at natural shifts. So we're going to be looking at small delta value changes from a negative number, say -25 to -22, and that's going to show that it became enriched. So now the next technique that I'm going to talk about is a technique that we can use to also prove biodegradation. But the delta values we're looking at for this technique are going to be very different, and I'm going to go through that and explain that to you. This is a tool called stable isotope probing. So as we just discussed, we know that in nature, 99% of carbon is carbon-12, 1% is carbon-13. But if we flip that ratio and we synthesize a compound such as benzene that I've shown here, where it's 99% carbon-13, it's going to behave the same as our original compound in the environment and the way it biodegrades, but then we can use it as a tracer to help us understand our contaminant fate. So let me walk you through this process. The way that it works is that we'll have a specially synthesized compound made. So it could be benzene like I'm showing here, it can be naphthalene, it can be MTBE or TBA, 1,4-dioxane, anything that the organism uses as an electron acceptor, we can put into this type of format. And then what we're going to do is we're going to load that compound onto our biotrap samplers, and I'll explain in just a moment what a biotrap is. But we can use that then to track the contaminant fate. We're going to put it into the monitoring well, one of the biotraps into the monitoring well for 30 to 60 days, and then remove it and do analysis in the lab. So before I get to the analysis of what we're going to do, let me explain a biotrap for those of you that may not be familiar with it. Biotrap samplers have been around for a long time. They're a passive sampling tool that was developed by the University of Tulsa and DuPont. And it's a way we can collect microorganisms so that we can understand biodegradation potential. The real key to the biotrap, you see those little units here in the picture. These are slotted PVC housing. The key are these little beads that are about the size of a BB. They're called bioset beads. This is what was developed by DuPont in Tulsa. They're composed of 75% powdered activated carbon, so compounds absorb very well and very tightly to these biotraps. And then the other 25% is material called Nomex. It's what fire retardant suits are made from. So we can deploy these in a monitoring well. They don't have any fossil biomarkers, and they've got a lot of surface area inside the beads. So we absorb these compounds on our biotrap in the stable isotope probing process. We're going to put like a 13C benzene on here, put it in our monitoring well. Then any organisms that are native to our monitoring well that can use this compound are going to colonize, form biofilms on these biotrap samplers, and then start to use that food source that we put down in the well. So the way that we're going to understand this is when we get the biotrap back from that monitoring well, we're going to measure the amount of that 13C compound that we have left. So we know what the loss was because we know how much we put on there originally. We look at what's remaining on the trap. Then, more importantly, not only was it lost, but where did it go? So we can look for our 13C incorporation into our dissolved inorganic carbon. So did the organism mineralize it and blow it off as carbon dioxide? Or we can look at biomarkers like our phospholipids, the cell membrane of a bacterial cell, and see did they make new biomass with this heavy-weighted carbon. So we can track and definitively prove that these organisms used this carbon and energy source that we put down in the ground. So I want to go through and kind of go through a quick example of a site where this was used and was very beneficial. This is a typical manufactured gas plant. It's been in operation for a long time. They removed a lot of the source area contamination, but they still had some naphtha present. The main contaminants were benzene, ethylbenzene, and some naphthalene that were left. The contaminant concentrations in geochemistry were suggesting that biodegradation was occurring at this site, but the regulatory agency wanted further proof of the biodegradation. They were asking for a third line of evidence. Also, they wanted to make sure that not only were the right microorganisms there, but they wanted proof that the organisms were utilizing those contaminants of interest. So we could really make sure that monitored natural attenuation was going to be an effective remedy for the site. So to answer these two questions, we used stable isotope probing to help us give that definitive answer about biodegradation, and we coupled it with some Quantaray and qPCR techniques to help us understand the concentrations of the microorganisms that are present. So here's the map of the site with naphthalene and methylnaphthalene concentrations. And first I want to draw your attention to well 7C here. This is our source area location, and it's really what I'm going to focus on for the talk today. We also have a down gradient location, and we're going to look at some Quantaray results on that. And then a background location here, 17D. So due to time today, I'm just going to show you just a little snippet of the data from the study. I'm happy to talk to anyone about it offline if you would like more information. But I'm going to start with first our stable isotope probing data. So for this study, we put a biotrap sampler that was loaded with a 13 carbon naphthalene compound into our monitoring wells. And we left it for a period of 60 days, I'm talking about monitoring well 7C in our source area. And what we see here is from the carbon dioxide, we see enrichment with our 13 carbon. So when we look at our background, DIC, carbon dioxide in the environment, the delta value for that would naturally be around a -25. But look at these values, we're well over 200. There's no way it could be that positive unless the microorganisms were producing carbon dioxide from that 13C naphthalene that we put into that monitoring well. So what about biomass? This is doing the same thing where we're looking at the delta values of our organisms so that we know that the phospholipids for organisms in the environment are typically negative, around, again, -25 to -35 delta values. But look at this, we're greater than 800 delta values, and there are times we see it into the thousands where they're just making a lot of biomass. So this is proof positive that the organisms were mineralizing the naphthalene, and now we're seeing proof positive that they were using it and making new biomass as they were growing, so incorporating that into their biomass at the site. So now I want to show you the Quantaray results for these same locations. So again, remember 17D is our background. Look at that. When we look at Quantaray, again, just focusing on the PAHs due to time today, we have no detection of aerobic or anaerobic genes for PAH degradation in those uncontaminated areas. But in the contaminated source area well, this dark blue, you can see we have both aerobic and anaerobic potential that's there, and particularly in that down gradient location, we've got a lot of anaerobic potential. And this gene that's circled here is an anaerobic naphthalene carboxylase gene that was identified a few years ago. The concentrations of this gene were around the 97th percentile when we looked at thousands of sites around the world. So very high concentrations of this gene that were present. So again, with this tool, we're seeing that the organisms are there. There's a lot of metabolic pathways that are available for the degradation of these PAHs, and then the stable isotope probing helped us to prove that degradation was happening at the site so that the site could get a ruling of M&A. So when they were looking at the assessment, the first line of evidence, the chemical line, was showing a green light, decreasing contaminant concentrations, and then when we used our additional lines of evidence from the microbial side, we saw with stable isotope probing, again, green light, things are looking good, biodegradation was proven, and then we see both aerobic and anaerobic pathways for contaminant degradation. So it was a really exciting for the site manager at this project to get this data because the regulatory agency was really pushing for them to do an ISCO type approach. And they were going to have to spend a very large amount of money. I think it was well over $1 million to implement an ISCO application. And in the studies that they were doing with the ISCO, they were seeing that it just wasn't going to be effective. They were only going to get about around a 10% reduction in contaminant because it's been around so long and the contaminant concentrations were low. So these tools help the regulatory agency to give them the approval to go with M&A so they could reallocate the money that they would have had to spend to other sites and other cleanup goals. So I think this is a very effective strategy. There's a lot of strength in doing stable isotope probing. Hopefully, as you saw from this example, it can give us conclusive evidence of biodegradation. With this type of technique, we don't have to know the organism or the pathway like we do with qPCR and Quantaray. We're looking at anything that incorporates that carbon into its biomass. And it's broadly applicable as long as it uses a carbon and energy source, we can do it. Now, the cost of this technique varies depending on the size of the contaminant. That's one of the questions I get a lot. It's inexpensive for common contaminants that are smaller molecules like the ones I have listed here. When you get into very large molecules, the cost goes up because the synthesis cost for the company that makes these for us is a lot more. Yeah, and like Alistair's saying here, I think this is the most conclusive data that you're going to see. You know, there's not a lot to interpret. It's really solid. When your numbers are that positive, there's no way that could be happening naturally unless the organisms were using that compound we put down. So it's really definitive proof for us. And before I move on to the final information we're going to cover today, we can use stable isotope probing in situ microcosms I'm getting ready to cover, where we can look to see what would happen under natural conditions. What does your incorporation, your delta values look like compared to, for instance, we added an ORC. Let's say we made oxygen bioavailable to these organisms. We can see, you know, kind of what difference there would be in the growth of our microorganisms if we added some type of electron acceptor to our site. Okay, so any questions about SIP or CSIA before I move on to our final topic? Okay. The last thing I want to talk to you about today is a tool that I'm also very fond of. It's called an in situ microcosm study. These are slotted PVC that you can see here in this picture. And we can use these as part of our biotraps to help us understand what treatment strategy should be selected. So we can set up attach different units together and deploy them into a monitoring well so we can understand what would happen under our natural conditions versus if we added different electron donors or acceptors. So let me zoom in so we can look at this a little more closely. Typically in an assembly, you're going to have a control unit that simulates natural conditions and then different treatment options. These are slotted PVC pipes. They're typically about 15 inches in length, depending on what we put inside the unit, so we can customize these depending on the questions we're trying to answer. The key to these is they have baffles on each end. So when we put them in a monitoring well, we can kind of seal off portions of that monitoring well and minimize crosstalk between the units. So we can really look at the chemistry, the geochemistry, and the microbial community within each one of these samplers. So inside, these kind of screw apart. It's like loading batteries into a flashlight. You're going to have some type of delivery mechanism for our electron donor acceptor. If it's a liquid, like a vegetable oil or some type of lactate or things like that, we're going to use typically a sponge as a supplier. If it's a powder, like an ORC, we would use a baggie to help deliver that. Then the other thing we're going to have is a passive diffusion bag, so we can look at what our contaminants of concern, what those concentrations are. We're going to have a biotrap with those little beads in it so that we can look at our microbial community. And then we're going to have a vial so that we can look at the geochemistry. And a good question that just popped up on this is, can we get in situ microcosms for 1-inch wells? No, unfortunately, it needs to be a little bit larger in diameter because we need to get enough volume to do all these different types of tests. And so to get enough volume for the geochemistry and the passive diffusion bags, we really need, I'm here in the US, it's a 2-inch well. So if we can get that or larger, that's what we would need for these types of studies. So we're going to have, as a result of these studies, you're going to get your chemical, your geochemical, and your microbial lines of evidence all in one little unit here. So let me walk through a quick example for you. This is a typical chlorinated site that's impacted by TCE at this site. DCE was detected in the groundwater monitoring data, but they weren't showing any vinyl chloride or ethane. So the client wanted to answer the question if biostimulation with an electron donor was going to be sufficient, or if they needed to do bioaugmentation. So the in situ microcosm was constructed with 3 units to give us the answers to those questions. The control unit will tell us if we have Dehalococoides under natural conditions. The biostim will show us if Dehalococoides responded, and then bioaugmentation, we can actually put a culture of Dehalococoides onto our bioset beads prior to deployment, and then we can see, will that culture actually survive those in situ testing? From the chemical side, we can answer questions like, what does our contaminant concentration look like now? Do we see daughter product formation? Are we seeing ethene, full reductive dechlorination in the biostim? And how much more do we see when we look at bioaugmentation compared to the biostimulation? So I just want to quickly walk through these results with you. This is looking at Dehalococoides in our blue bar. The red and green are Dehalococoides genes, TCE reductase or vinyl chloride reductase. You can see under natural conditions, we have very low concentrations of Dehalococoides that are present. Post a 60-day deployment, we have more than an order of magnitude increase in our Dehalococoides concentration. So we're not to that 10 to the 4 mark that we've talked about where we want to be, but I'm really encouraged when I see that number, because in a 60-day period, if we can get fermentation of that electron donor that we've added and see Dehalococoides be stimulated by more than an order of magnitude, it's pretty indicative that that's going to be even more successful when you go to a full-scale application and you have a longer time for that process to happen. With the bioaugmentation unit on the microbial side, what we're seeing here is that we have 10 to the 7 cells of Dehalococoides that are being detected. We know pre-deployment, 10 to the 7 is what we loaded on there. So if we recover something similar, to those initial concentrations, it's a good indication that our Dehalococoides population can survive in situ. And just as a side note, there have been several studies that I can think of where we did this and the bioaugmentation wasn't successful. The conditions weren't right, and we saw Dehalococoides populations plummet during that 60-day deployment period, where we'd recover maybe 10 to the 4, even one study I remember 10 to the 3 cells of Dehalococoides post-deployment. So this is a great tool to help answer the question about the survivability of the culture. So from our microbial lines of evidence, what we learned is that we have Dehalococoides there, but it's low. We are seeing a noteworthy increase in these organisms. Do we need to do bioaugmentation? Possibly not. We can talk about this more after we look at the chemistry and talk about the site goals, but it's a pretty good stimulation that happened. And then will the culture survive? Yes, we were able to see it did survive during this study. So let's look at the chemistry from our samplers. Plotted here shows the TCE and cis-TCE, or the primary contaminants being detected in our M&A unit. Look at our biostimulation unit. We enhanced the Dehalococoides, gave them some electron donor. We're starting to see vinyl chloride and ethene formation. So in a 60-day period, it was pretty exciting to see that happen. The bioaugmentation side, we do see more daughter products that are being formed, but it's not a huge difference. So at this point, the site manager can start to make a decision. I've got biostimulation that's happening. I got enhanced DCE production, seeing full reductive dechlorination. So what's my receptors look like? What's my time look like? So they can then decide between biostimulation and bioaugmentation based on those types of questions. In this particular situation, the client moved forward with biostimulation. They didn't have an immediate impact they were concerned about, so they had a little more time with this, and so they got to save a little bit of money and not move forward with the bioaugmentation, just do biostimulation. So as I wrap this up on this tool, I think in situ microcosms are a great tool, whether we're talking about chlorinated hydrocarbons or petroleum hydrocarbons. It can help us answer a lot of questions that we would try to typically do on the bench scale, but doing them in situ. And while it's not a replacement for a pilot study, we can't do all the things we can in a pilot study, it's a good intermediary and it's a lot less expensive. So if you're screening different remediation options, this is a great tool that you can use for that. So before I end today, I know you're probably tired of hearing me at this point, but I wanted to wrap up with just a little bit of information about Microbial Insights. Microbial Insights has been around for a long time. We were established in 1992 as a technology transfer from research that Dr. David White did at the University of Tennessee. Dr. White was a real leader, very innovative, always thinking about how do we transition tools from the medical field to the remediation industry so we can better understand our sites and do cleanup. So it's our goal at Microbial Insights to continue his legacy, to use that experience and be innovative, to offer the most comprehensive suite of everything that we can, and to be very customer service oriented so that you guys can get what you need in a efficient time. We've been tested numerous times against labs around the world, and every single time Microbial Insights has been kind of to be the best lab that was the most accurate. We are very serious about QA/QC. Happy to share any of that information if anybody has questions about it. We do have our lab there in Adelaide. Love the lab there. They do a spectacular job, so it makes it a lot easier on the shipping end for you guys to get it over to there. HydroTerra is a great resource there for you guys in Australia. And if there's a question they don't know the answer to, they're great to get it over to us and we'll make sure that we respond. I would encourage you, if you have questions, contact me, contact Kate or Casey that I've listed here. They're two of our project managers, very good. They can help you with anything that you need about a site. I would encourage you not to let the amount of tools or the amount of information I've shared with you today be overwhelming. if you call us and say, you know, Dora, here's my contaminant, here's what I'm trying to do at this site, the questions I want to answer, I can try to help answer those questions for you. And the Microbial Insights website, I think, is a valuable resource for you. You can actually copy and paste a lot of information into your proposals from there. And if you want to call, we can always set up a call to talk to you guys. So I guess at this point, I'll take any remaining questions, or if there's anything else, Alistair, that you think I need to cover, I'd be happy to.
No, that was fantastic and really comprehensive. Thank you very much, Dora. I will reiterate that about what you were saying with, I suppose, the customer service about deployment, and Microbial Insights are always give a lot of time in advising and helping to find the best course of action for particular sites. If anyone has any questions, you can either type them into the chat box there, or you can. There's a little raise your hand button, and I can give you audio if you'd like to ask a question. I have a question that I'm going to immediately regret. Actually, I think. Just thinking about the 1-inch wells. Is it possible to sort of do a pump sampling of a well and make a little artificial environment to do the in situ microcosms in? Sort of make a sort of in situ environment.
I suppose you could, but I feel like we might be getting back to a bench scale test at that point. We really want to be able to leave it for a long period with some natural flow, so that we get the recharge that would happen within the unit.
Yeah.
So if any other option, I think that would only be a backup mechanism for us.
Okay, so I don't have to fully regret my question. Jeremy has a question, so I'll just pass the microphone over to Jeremy here. I'll just, if it's all work. There we go. Jeremy, you might need to, yeah, no problem. Do you want to type your question in into the chat box, maybe? That will work. I've got another one while you're typing it in. So I was thinking with the SIP, it is really sort of irrefutable evidence, and I was thinking about an auditor and where they might sort of come up with a problem, and it might be around sampling. Is it just the sort of normal sampling guidelines required for a site, or do you recommend any other specific sampling guidelines for this?
You know, for stable isotope probing, we would send you a biotrap that's preloaded with the contaminant of concern, and most compounds bind really well to the pack, so we don't have to worry about any leaching into the monitoring well. That's not typically a concern. We also recommend that you deploy them in wells that have contaminant. So if, you know, there was something strange that happened, the amount of contaminant on there is very low, so it wouldn't really impact any of your groundwater monitoring. But that's really the only thing there is to it. It's to put this biotrap in the monitoring well, leave it for 30 to 60 days, depending on the process we're trying to target, and then simply ship it to our lab here in Adelaide, and we'll do all the analysis.
Mm-hmm. Yeah, it's a really attractive option. I think Jeremy's still typing his question out, so I'll just pause 1 second. Does anyone else have any questions? Nick and Sam, who are online, I should say as well, Jeremy's question's there, but I'll just point out that Dora and I recorded a fascinating podcast that talked about some of the technology from Microbial Insights, but also a bit of background around about the company, and also some insights into, no pun intended, some insights into where technology might be going in the future as well. So I'll include a link to that podcast in my email after this. But over to you, Dora, with this question.
Okay. All right, thank you, Jeremy. That's a good question. So I think you could probably all see that. With remote sites, it can be difficult. If you freeze it, my key would be it needs to stay frozen until it gets to our lab. We don't want to let it freeze thaw because you can start to shear the DNA. So if you freeze it, you just got to keep it frozen and make sure you ship it so that it can get there still frozen. Kind of in those situations where you're talking weeks or longer, freezing is probably your best option. So I would recommend when you ship it, do it on dry ice. Make sure your container is sealed very well so that it can stay frozen during that transit. What you're at. I would prefer that you keep it at -80 if possible. -20 will work. That just, the colder we can get it, the less chance of it freezing or less chance of it thawing during transit.
Excellent. Thank you, Jeremy. Thank you, Dora. If there aren't any more questions, we'll leave it there. So a final thank you to you, Dora, for your time. I know it's early evening over in the US at the moment, so I really appreciate your time putting it aside for this.
Happy to do this, and I'm happy to send a PDF of the slides if anyone wants it. And I think you were recording, is that right, Alistair?
Yeah, that's right. Actually, if you could send me those PDFs, that'd be great. And I'll put it together in a whole package. So there'll be audio, the recording, the slides, as well as the link to the EPA guidelines, as well as the podcast on there.
Sure, I'll be glad to do that.
Thank you, Dora. Thank you, everyone who's still online. That was absolutely fantastic. I look forward to seeing everyone maybe at our next webinar, and I look forward to catching up with you soon, Dora. Thank you very much.
Thank you.
All right, bye now.
Bye-bye.